everyone, welcome to our online lecture. Today, we will be talking about the coupling of chemiosmosis in electron transport chain and in ATP synthase. Basically, chemiosmosis is the movement of ions across semi-permeable membrane down their electrochemical gradient. An example of this would be the generation of adenosine triphosphate ATP by the movement of hydrogen ions across a membrane during cellular respiration or photosynthesis, while ETC is a series of complexes that transfer electrons from electron donors to electron acceptors via redox reaction and couples this electron transfer with the transfer of protons, hydrogen ions across a membrane. We know that this whole process occurs in mitochondria. There are three important parts of mitochondria we have to know here. This is the intermembrane space, the inner space, and this will be the matrix of mitochondria. So, this part here is the ETC and this is the oxidative phosphorylation. So here is the membrane and within it is a bunch of protein complex. They are known as the complex 1, NADH dehydrogenase, complex 2, succinate dehydrogenase, complex 3, cytochrome C reductase, complex 4, cytochrome C oxidase, and the last one is the ATP synthase, or sometimes known as complex 5. It is a complex that makes the ATP. The other factors involved in the electron transport chain are the coenzyme Q10 and the cytochrome C complex.
So, the process begins with NADH, which is produced in glycolysis and TCA cycle. First of all, NADH from glycolysis is produced in cytoplasm. This NADH can diffuse from the outer mitochondrial space into the intermembrane space of mitochondria, but it cannot diffuse through the inner membrane to the matrix. So, this NADH will pass electron to shuttle. In the matrix, NADH is oxidized to NAD+, and it donates 2 electrons to the electron transport chain. These 2 electrons will get passed between this complex like this. And this electron will finally be accepted by oxygen. And with a couple of protons, this produces water. This oxygen here is supplied by breathing from our atmosphere. This explains the reasons why animals need to breathe. Now, as the electron pass between these complexes, they pump protons out of the mitochondrial matrix into the intermembrane space. Complex 1 pumps out 4 protons, complex 3 pumps out 4 protons, and complex 4 pumps out 2 protons. Therefore, in total, 1 NADH will produce 10 protons. A similar process occurs when FADH2 is used as the electron donor. 2 electrons will get donated to protein complex 2, which will then pass through coenzyme Q and onward down the electron transport chain. Because complex 1 is not involved in FADH2 as the electron donor, only complex 3 and 4 pumps out electron, and that would make a total of 6 protons. Finally, these protons will then flow back through the ATP synthase, and 1 molecule of ATP is produced. To sum this up, for NADH as the electron donor, a total of 2.5 ATP is produced, while for FADH2, only 1.5 ATP is produced. So, that's it for today. Thanks for watching, and I hope you enjoyed this video, and good luck for final exam. Okay, bye.
Okay, now let's look at the effects of respiratory inhibitor on ETC. Okay, respiratory inhibitors are substances that bind to some components of ETC thus blocking its ability to change in a reversible form from an oxidized state to a reduced state and vice versa. In this case, different inhibitors bind to different components in ETC. Amytal and rotenone bind to complex 1, prevent NADH from being utilized. However, ATP can still be produced since the electron flow from oxidation of FADH2 in complex 2 is not affected.
Another inhibitor is antimycin, which bind to complex 3. Cytochrome BH in complex 3 cannot be oxidized, thus interfere the electron flow. Cyanide, azide, and carbon monoxide are inhibitors that bind to complex 4. Cyanide will bind to Fe3 plus ion, which is an electron transporter in complex 4. Inhibitor azide act in a similar way as cyanide. Carbon monoxide will bind to hemoglobin, forming carbaminohemoglobin, interfere the transport of O2 into the cell. When there is no oxygen molecule to receive electron transport change cannot be completed.
Created using Powtoon.